Hello, and now we're moving on to 9.2, the process of cellular respiration. Cellular respiration in more details. So the goal is to identify the reactants, products, and basic functions of aerobic and anaerobic cellular respiration. Far side says, in what was destined to be a short-lived spectacle, a chicken suspended by a balloon floated through the samurai bar's doorway. Just something a little silly. The objectives are all about cellular respiration. Describe what happens during glycolysis, describe what happens during the Krebs cycle, explain how high energy electrons are used by the electron transport chain, and identify how much ATP cellular respiration also generates. So yes, food does burn. In fact, carbohydrates found in flour and protein in flour can explode, and this has caused several explosions in flour factories. I don't have a picture of it, but it's more fun. Plenty of energy is available in food. So how does a living cell extract energy stored in food without setting on fire or blowing things up? Well, let's start with the glycolysis. This, by the way, is all the details in the cellular respiration. If you really want to get into it and study it, go right ahead. But we're going to do a little more broad overview here, even though it's going to seem like a lot of details to you. You can see it can get much more intense. All right, let's start with glycolysis. Glycolysis is the first stage of cellular respiration and occurs in the cytoplasm of the cell. During glycolysis, the glucose, right here, is broken down into two molecules of three-carbon molecule, right here, 3 carbon molecules of pyruvic acid. Pyruvic acid is a reactant in the Krebs cycle, so that's going to go on to the Krebs cycle and be used there. ATP and NADH are also produced, as you can see. So let's see what happens. The cell deposits 2 ATP, right here, 2 ATP molecules into an account, basically, to get things going. This is like the investment phase. So it has to use 2 ATP to get glycolysis started. The phosphate that is then released, right here, the phosphates from the ATP that's released off, actually binds on to the 6-carbon chain, and it breaks it, and also is used, the energy from that is used to break this 6-carbon chain into 2 3-carbons with the phosphates on the end, and these are called PGALs, phosphoglyceraldehyde. Ugh, that's a mouthful. So then, glycolysis then produces 4 ATP molecules. All right, more energy is being used by that, giving the cell a net gain of 2 ATP for each molecule of glucose that enters glycolysis. So it's 2. With these, you're going to get 2 here and 2 here. All right. Also, during glycolysis, the electron carrier NAD+ accepts a pair of high energy electrons and then becomes NADH. So again, we have some energy being stored here. The NADH carries the high energy electrons to the electron transport chain, which comes after the Krebs cycle. All right. So this, you're going to see it again, where they can be used to produce more ATP. 2 NADH molecules are produced for every molecule of glucose that enters the glycolysis. So, glycolysis produces ATP very fast, which is an advantage when the energy demands of the cell suddenly do increase, like you're trying to sprint really quick and you're not getting enough oxygen, because glycolysis does not require oxygen. So it can quickly supply energy to the cells when oxygen is unavailable. So, glycolysis, what you're going to need to use in glycolysis, the input is glucose, 2 NADH, it's 2 NADs, 2 ATPs, 4 ATPs, and 2 phosphates. All right, when they go through the glycolysis, what you're going to get back are 2 pyruvates, 2 NADHs, 2 ADP, right, because we used 2 in the beginning and now they become ATPs, and then you're going to get 4 ATPs. So your net product then is your 2 pyruvates, 2 NAD+, and 2 ATPs. If you want more detail, click on the Miller-Levine video. All right, let's take another look here of glycolysis. You have your glucose, some ATP is being used here. All right, we have our phosphate group is being broken down. All right, this is just one of them. Here it gets broken into 2 carbon chains. You can see there's oxygen in there. Oxygen is attached to the glucose and the phosphate, but you don't need oxygen for this reaction to happen. You have your NAD, that becomes NADH. It's going to click on another hydrogen ion there. All right, and again, this happens twice. So this goes this way, and then this reaction goes over that way. So this is follow one of them. ADP is being used, or it's become ATP, energy is being made there, and then it just goes through a series of reactions, some water is produced, and then again, you can look at this in more detail. I do not want to spend too much time right now when we're going to be practicing in class. Otherwise, your video will be extremely long. Okay, objective 1 was describe what happens during glycolysis. So during glycolysis, 1 molecule of glucose, a 6-carbon compound, is transformed into 2 molecules of pyruvic acid, which is a 3-carbon compound. Moving along here. Objective 2, into the Krebs cycle. All right, during the Krebs cycle, the second stage of cellular respiration right here, pyruvic acid produced over here in the glycolysis gets oxidized and is broken down into carbon dioxide in a series of energy extracting reactions. The Krebs cycle is also known as citric acid cycle because citric acid is the first compound formed in a series of reactions. Happens right about here, citric acid. All right, let's move on though. So, the citric acid production of the Krebs cycle. Pyruvic acid from glycolysis, all right, enters the matrix, which is the innermost compartment of the mitochondria. All right, enters inside through this membrane, in through this membrane and inside of it. All right, this is where it's happening. So once pyruvic acid is oxidized, which means it's going to lose a hydrogen, keep that in mind, those hydrogens are going to build up again like in photosynthesis in the mitochondrial matrix in here. NAD plus accepts 2 high energy electrons to form NADH. Okay, so NAD plus is then reduced to become NADH. Why is it being reduced? Because it's accepting electrons, and electrons are negative, so it's being reduced. Even though it takes something that sounds different, it's backwards. But so 1 molecule of CO2 is also produced. This is where we're getting our exhale stuff from. The 2 remaining carbon atoms react again. The 2 remaining carbon atoms react to form acetyl-CoA. All right. So acetyl-CoA is a 2-carbon molecule, combines with a 4-carbon molecule, which was already present because remember it is a cycle. All right, to produce citric acid. The citric acid is then broken down or oxidized. Why is it oxidized? Because it's losing, all right, into a 5-carbon compound, and then more CO2 is being taken out, and then it gets oxidized to a 4-carbon compound. Again, more CO2 is being released there too. The 4-carbon compound can then start the cycle again, all right, by combining with acetyl-CoA again, up there. So, energy, what's happening when this is going on? Energy released by the breaking and rearranging of the carbon bonds is captured in the form of ATP, yay, NADH, yay, and FADH2. Something different for you there, but it still has energy. That's what you need to know there. So, for each turn of the cycle, 1 ADP, oh, why I didn't fix that? 1 ADP molecule is converted into ATP and can directly power the cell's activity. All right, the electron carriers, NAD plus and FAD, each accept pairs of high energy electrons to form NADH, like we said, and FADH2. NADH and FADH2 are used in the electron transport chain to generate ATP. So again, these guys, see the arrows, go down to the electron transport chain, down to the electron transport chain. They go down to the next level. All right, so this is how this energy is going to happen in an electron and how we're going to get a lot of our ATP. Remember, each molecule of glucose results in 2 molecules of pyruvic acid, right? 2 of them. Remember, it was doubled, which enter the Krebs cycle. So each molecule of glucose results in 2 complete turns of the Krebs cycles. Therefore, for each glucose molecules, you get 6 CO2 molecules. You got 3 here, but double it, you get 6. 2 ATP molecules, 8 NADH molecules, and 2 FADH2 molecules are produced. So, yeah, sorry, I forgot to click. So anyhow, here's the totals here. Krebs cycle totals 4 NADH in 1 times 2 is 8, 1 ATP times 2 is 2, and 1 FADH2 times 2 is 2 FADH2s. All these things get to go down to the electron transport chain. Again, more information on 442. All right, so our goal was to describe what happens during the Krebs cycle. During the Krebs cycle, pyruvic acid is broken down to carbon dioxide in a series of energy extracting reactions. And here it is, AP style. Same thing. You can see the reactions happening right here. CO2, CO2, NADH, all that. Take a closer look when you're ready, all right? Okay, objective 3. Explain how high energy electrons are used by electron transport chain. This is where all these stuff went to, right? Okay, so NADH and FADH2 pass their high energy electrons to the electron carriers. Look at that right there. Electron carriers, proteins, and the electron transport chain. These guys. Cool. At the end of the electron transport chain, the electrons, this would be the end over here. The electrons, I should click, sorry. The electrons combine with H plus ions and oxygen, all right? H plus ions and oxygen, because these things build up in there, and oxygen. These electrons all combine, and you get water. Yay! Energy generated by the electron transport chain is then used to move those hydrogen ions against the concentration gradient again, just like photosynthesis, across the inner membrane, okay, across the inner membrane into the intermembrane space. Okay, this is between the cristae and the membrane of the mitochondria. These little hydrogen ions are being built up. So the hydrogen ions pass back across the mitochondrial membrane, all right, right here, through the ATP synthase, these guys right here, causing the AT synthase, just like photosynthesis, molecule to spin. And with each rotation, the ATP synthase attaches a phosphate group to the ADP that were left around, floating around before because they were used, remember, to produce ATP. There's an interactive video on page 44, by the way. So the inner foldings of the cristae, all right, the reason that happens, why it's all bunched up in there like that, is because of the membrane surface availability. The more it can squish it up in there, you know, in a ball, whatever, the more you have electron transport chains going on in there. So it doesn't happen in one spot, it happens all over the mitochondria cristae inner membrane there. Hey, cool, huh? So, objective 3 was explain how high energy electrons are used by electron transport chain. All right, the electron transport chain uses those high energy electrons from glycolysis and the Krebs cycle to convert ADP into ATP. Now, how much ATP did we get from cellular respiration? How much did it generate? All right, the energy totals. In the presence of oxygen, the complete breakdown of glucose through cellular respiration, gotta have oxygen, results in the approximate production of 38 ATP molecules. This represents about 36% of the total energy of glucose, the original glucose that went in. The remaining 64%, guess what? Heat. And we keep our bodies nice and warm with it. All right? So the cell can generate ATP from just about any source, even though we modeled it using only glucose, all right? Complex carbohydrates, there we are broken into simple sugars like glucose, lipids and proteins, lipids and proteins and fats, lipids are fats, proteins, all right? It can be broken down to molecules that enter the Krebs cycle. They don't go through glycolysis at one of the several places. And some can go, but most don't. All right? All right. There's a tutor 2 video on this on page 445 in your book. Also, crash course. If you haven't seen this guy, he's really funny and he has a good video on cellular respiration too. Objective 4, identify how much ATP cellular respiration generates. So together, glycolysis, the Krebs cycle, and electron transport chain release about, about, not much more, but could be less, 38 molecules of ATP per molecule of glucose. So glycolysis, you get 2 ATP and 2 NADHs. Krebs cycles to 2 ATP, 8 NADH, 2 FADH2s. And then so far, anyhow, you can add it up. I don't want to waste your time. 38 ATP is what you need to know. All right, so ask yourself, you probably can't if this is your first time looking at it, so you need to go back and study it really well and look at some more videos. Khan Academy has something too, but make sure you can describe what happens during glycolysis, describe what happens during the Krebs cycle, explain how those high energy electrons are used by electron transport chain, identify how much ATP cellular respiration generates. And there's my puppy. That's really what he looked like when he was a baby. That's not him, but he was so cute. I love him. Bye.